Okay, great. Okay, you can go ahead and put up the first slide. Great. Okay, those were two great talks, so this is a lot to follow. So what my lab is interested in doing is elucidating bacterial virulence factors that bacteria use during an infection, and we try to understand the molecular mechanisms that they use, and it's kind of a detective story for everyone because there's not a lot of information provided about the virulence factors before we start studying them. But to try to put this in context so that all the different types of scientists in the audience can understand what we do, I'm going to use an analogy that had been used earlier, and that is a house. So you can think of a house as like a cell in your body, and its similarities are that a house determines its shape dependent on the frame. So when you build the frame, then that's going to tell you what the shape of the house is. In our cells, what we have is a membrane, and inside we have this thing called the actin cytoskeleton, and that's very similar to the frame in the house, and that dictates the shape of the house. Now, if you have a house and you have termites in your house and you destroy the frame, then what's then going to end up happening? Your house will collapse, right? And so that's a very bad thing to happen. The other thing that's in your house that is very important that we use a lot of are switches. So we have binary switches in the house to turn on and off the lights. We have dimmer switches. We can make them on a little bit and turn them on higher. Another really important switch that we have in our house that everybody knows in July and August, they go home and they basically crank this guy up. They crank their air conditioner on so that they can cool down the house and make the environment very nice. And there's another switch in the house, and if this switch is lost, all hell breaks loose because it's the remote control to the TV. And everybody knows that this is an important switch because it can control volume and channels, and it has a lot more buttons on it than we actually need, and I get confused sometimes. But it's a very important thing for maintaining nice homeostasis in the house. And then, of course, we've got a sink. We can turn on the water and turn off the water, make the water hot, make the water cold, increase the volumes and decrease the volumes. So imagine your house is attacked by a pathogen. You go home one day and you walk into your house and the thermostat doesn't work. And it's July in Texas, okay? The house is very, very hot. It's very uncomfortable. And then you walk in your house and you're walking around and you realize all the lights in your house are on. And you can't turn them off. Something has permanently turned on all the lights in your house. And this is making the house even more warm. So you decide you're waiting for somebody to come repair these things and everything, so you're going to distract yourself. And of course, what has happened? Someone has lost the remote control to the TV. So you have nothing to distract yourself. So this has just wreaked havoc in this place where you're supposed to go home and feel comfortable. So I'm using this as an analogy because this is kind of what happens when a bacteria comes and infects your cells. It basically comes in and takes over switches, and it determines whether the switches will be on or they'll be off, or they can destroy switches, or that you completely lose control of what's going on. And they manipulate the situation for their own advantage during an infection. So what I propose is that by understanding how the bacteria work, we can use this as a model for discovery. So what I've told you is that during an infection, bacterial pathogens disrupt and deregulate molecular switches in our body. And based on this hypothesis, we have done the following. We've uncovered the activity of bacterial virulence factors, and in doing so, we've not only understood switches that we already knew about, but we've discovered new switches. And so today what I'm going to tell you about is I'm going to go through two stories about two different switches that actually you heard about last night, how we can learn about how our bodies work by understanding these new molecular switches that we discovered through the bacterial pathogens. So the first thing that I want to tell you about is how these bacteria that I study work. So they're extracellular pathogens. They stay outside of the cell that they're working on. And in this case, what you're seeing is a bacteria nestled against a macrophage. And they have this really cool tool. It's called a type 3 secretion system. And so what they do is these little colored boxes in the bacteria, those are designating virulence factors. And the virulence factors are injected through this needle-like structure so that you have a direct communication between the inside of the bacteria and the inside of the cell that it's infecting. And when they go in, what they do is they go disrupt switches. They turn things on, they turn things things off, and they manipulate the cell to the advantage of the bacteria. And at the bottom, I have the kiss of death, because actually this whole system has been coined the kiss of death by Yersinia, because Yersinia pestis, which was the causal agent of the plague, uses this system. And when it injects these virulence factors, it kills the cell it comes in contact with. And it also manipulates signaling systems. And so the first effector that I'm going to tell you about today is this one called Yersinia. From Yersinia, it's called Yop-J. And so, and Yop-J carries out a very particular function during infection via this mechanism where it's taken from the bacterial cell and injected into our cells. And so you know that the plague was the causal agent of a third of the deaths in the Middle Ages, a third of the population dying in Europe from this bacterial pathogen. And so, it has to be very clever in what it does. And so one of the ways of thinking about what Yersinia does during an infection is you could think about it if it wrote an NIH grant. It would have specific aims, okay? So the pathogen goes in, and it's an extracellular pathogen. It doesn't want to be engulfed. It doesn't want to be eaten by our cells and destroyed. So it has to have a mechanism to evade engulfment. Another thing that it wants to do is wants to cripple the host immune system, because it's hiding out in your body and it wants to replicate, right? So it doesn't want to alert the immune system to say, hey, I'm here, come and get rid of me, okay? So it wants to hide. And then the other thing it wants to do in hiding, it wants to propagate. It wants to make more of itself, right? And if it propagates, and there's a lot of bacteria in an infected individual, it can become virulent, right? Because it's so infectious in that individual, it can be passed to an individual who isn't infected. And that basically provides the Yersinia to exist. So, we started studying an effector, this Yop-J, and what we discovered is that this effector is involved in crippling the host immune system. So this one protein that the bacteria injects into cells is required for decreasing our ability to sense that the bacteria are in our own bodies. And so we study these things and we look at these molecules, and basically what they are is a string of amino acids. And we say, how can we figure out what this thing is doing? And we have a lot of different tools that we use. And one of the first tools that we use is bioinformatics. So we looked at this molecule, and we looked at a lot of molecules that other bacteria that make that look like Yop-J. And we said, you know, this really looks like a protease. It's probably a hydrolase. So this is something that cuts up other proteins, right? And the reason we thought this is because it had a nice conserved catalytic triad. It looked like the type of enzyme that could do this. It had the right amino acids for this. And if we took the protein and we predicted the secondary structure and folded it up, it really looked like a protease. It looked like something that could cleave other proteins. And so we went on this hypothesis, and we started asking, well, what types of things does Yop-J do when we put it into cells? And what we discovered is that for the people that, for the biochemists and cell biologists in the audience, we discovered that this one molecule, when it's injected into cells, can block every single MAP kinase signaling pathway and the NF-kappa B pathway. And so what this means is those are all of the signaling pathways in your cells that will trigger the type of molecules that we heard about earlier that alert the immune system, okay? So all of those pathways are required, and this one molecule can block all of those. So that means the cell comes in, what usually happens in a cell is in this pathway, the signaling pathways are off. And then when the cell gets infected, these signaling pathway is turned on, and when it's turned on, it sends out signals to the rest of the body that I'm infected, that this cell is infected. And so what I've shown here is a molecule that's in the off state. In this case, it's the MAP kinase. And when it's off, it has no activity. When it gets turned on by another kinase, phosphates are added to this molecule on this S and T, which are serine and threonine residues. The phosphates are a negative charge, and they change the shape of the protein, and it's been modified. And changing the shape turns this enzyme on, and now it can go alert the rest of the cells that, you know, the body's been infected by a bacteria. What Yersinia does is it injects this molecule called Yop-J. And what we found is that Yop-J kept this signaling molecule in the off state. It's completely off. But it's different than the off state of an uninfected cell, because this molecule in the off state cannot be turned on. So, but we couldn't figure out how this was happening. And remember I told you we thought YopJ was a protease or a hydrolase. So we started looking at all the molecules involved in this pathway, and we said, what's being cut, what's being cleaved, what's changing in molecular weight, and we couldn't find anything. And so we kept muddling around with this, and then finally we decided, okay, we know this. We know the off state in an uninfected cell is different than an off state in the presence of YopJ. So what we're going to do is we're going to determine the molecular mass of those two molecules and try to figure out what YopJ might be doing to this signaling molecule. And when we did that, we got a really big surprise. And the surprise was that this molecule didn't decrease in molecular weight, because remember we thought J was this protease that would cut it. It increased in molecular weight. And it increased in increments of 42 Daltons. And this, based on our analysis, is equivalent to an acetylation, an acetyl group that's added to the serine and threonine. And this was a really, really cool result for quite a few reasons. The first is that J wasn't acting as a hydrolase now, it was acting as a transferase. And it was transferring to this signaling molecule an acetyl group. And the acetyl group is in the exact same place where the phosphate should be. Okay? So what we were observing is a competitive modification on a signaling molecule in a cell. And this is a completely new mechanism because no one had ever seen an acetyl group on a serine and threonine residue. Classically, they're only found on amines, which are on lysine residues or the amino terminus of, on the amino terminus of proteins. And so this was a really cool observation. And basically what we found is that J affected a switch in a cell, and it did it in a very unique way in that it permanently turned it off. But it not only turned it off, it put it into a state where it no longer could be turned on. And so that completely shut down the signaling in our cells so that we can no longer recognize that our cells were being infected. So what we discovered was that YopJ was an acetyltransferase, and it acetylates these critical residues on this signaling molecule, and that it inhibited the activation of these kinases by phosphorylation because those sites were taken up and they can no longer be modified. So then you're saying, you started out this whole thing telling me that this was a hydrolase, and now you're telling me it's a transferase. And how can you make sense of this? And the sense is made by the beauty of chemistry. It's absolutely beautiful because it turns out that hydrolases and transferases use the exact same mechanism, but they use different substrates. And to explain this, I've depicted it in the following way. So the same mechanism is a ping pong mechanism. So if you look at the E, it has a CYS on it. That's a cysteine residue. And that's the nucleophile that was in that catalytic site that I told you was working, we thought, as a hydrolase or a protease. It attacks the first substrate, acetyl-CoA, and in the second part of this E, it makes this acyl enzyme intermediate, which is really a high-energy intermediate. And then the second substrate is a protein, and the protein comes in and attacks that, and now you can see with the final panel with the E and the S that the acetyl group has been transferred to the S moiety. And that's basically how a transferase works. So how does a hydrolase work? Well, it works the same way, except it has a different set of substrates. So the enzyme that works as a hydrolase attacks, in this case, a protein, a peptide bond. And what you end up with, in the middle, you can see the purple E and the blue E have the same kind of linkage. Okay, it's an acyl enzyme intermediate. But the second substrate for a protease is water because it's a hydrolase. It's breaking a bond, right? And so the difference between these two enzymes, they look very similar by bioinformatics, but when you do the biochemistry, you realize it's a completely different reaction. And this was a really, really important lesson that we learned by looking at these virulence factors, because you can't really predict what they are until you figure out the biochemistry or the activity that they take, that they perform. And the other thing that we realized is that you can't necessarily predict that something isn't a transferase until you do this kind of biochemistry. And a transferase absolutely cannot have a water in its catalytic site, because if it did that, it would turn into an acetyl-CoA hydrolase, which would kill any cell that it was in. So this was a really cool discovery that we made, and it was one of the first effectors that we figured out through this kind of path where we had to do all of this detective work. And because we figured out this story, it actually has made other detective stories a little bit easier. So the second effector that I'm going to tell you about comes from Vibrio parahemolyticus, and if you've ever eaten oysters and gotten sick, it's the guy that probably made you sick, and that's what I call the black pearl. So this bacterial pathogen uses a similar mechanism to what Yersinia does. It has a type 3 secretion system, and it has all these little effector molecules get injected into a cell. In this case, it's probably the epithelium in your gut during gastroenteritis when you've eaten this pathogen. And what it has is a different repertoire of virulence factors that are injected into cells, and they do the same thing. They go in and they manipulate switches, they turn switches off, right? Or they turn them on, all to the advantage of the bacterial pathogen. And the effector that I'm going to tell you about today is, the second one is this one called Vibrio VopS. And VopS is a really cool little effector guy, because now what I want to do is I want to go back to the house. Do you remember the house that has a frame, and the frame dictates the structure of the house, right? And so on the left side, you'll see normal cells, and the red is basically this beautiful actin cytoskeleton, and it basically defines the shape of the cell. And what I'm going to tell you is that VopS is a termite, okay? It has gone in and totally destroyed the actin cytoskeleton in the cell, and what happens is all the cells round up, and then they eventually die. And so this molecule has had a terrible effect on the frame of this house. And so our job was to try to figure out how it was working. So we use the same types of tools, right? So we go and do bioinformatics, and we say, well, what does VopS look like? And it's this protein, and it has this thing called a Fick domain. And that's cool, but we don't know what a Fick domain is. Nobody knows what a Fick domain is. So we do a little more investigation, and we find out humans have a Fick domain, flies have a Fick domain, worms have a Fick domain, bacteria have Fick domains. Bacteria have tons of Fick domains, and they use them as virulence factors, and they use them probably as something to do in a normal metabolism based on bioinformatics. We predict these things. But we have no idea what this molecule is doing. So then we did some more investigations, and we said, well, what are important molecules in maintaining this actin cytoskeleton, the shape of the cell? And so we started looking at some molecules, and what we discovered is that VopS was impinging on this G protein signaling pathway. And these are monomeric G proteins, and they're RAC-Rho and CDC42, and in this case, I'm going to talk about RAC. So what happens is, in an inactive state, RAC is bound to GDP, and it has a different shape than when a nucleotide is exchanged and it's bound to GTP. And the way that this system works, it's a lock and key. This is how this switch works. So what happens is when RAC becomes active and it's bound to GTP, the key fits into the lock. Okay? And when the key fits into the lock, then you can make all this actin cytoskeleton, and the cells have this nice shape. But what we discovered is that what happens is during an infection, VopS is injected into cells, and it goes and it targets these little G proteins, and it inactivates them, and it does something to them. So now the key can no longer fit in the lock. Okay, so something has happened to this system. We thought, oh, well, maybe VopS is just binding to these G proteins, but bacteria injecting a very small amount of protein. And so we predict many times these mechanisms are probably going to be catalytic, right? And so it's doing something to this molecule so that it no longer can fit in the lock, and now the cells round up, right, because they can't form their actin cytoskeleton. So we said, okay, we're going to make a really easy assay. We're going to have an assay that whenever RAC is bound to PAC, we get a black band on a gel. And if it can't bind to it, then we don't get a band on a gel. Okay? So the assay is like this. So in the absence of VopS, if we take RAC and we do this assay, we get a black band on a gel indicating that the key has fit into the lock. Okay? If VopS is around, the key no longer fits in the lock and we don't get a band. So we looked at this and we said, you know, we don't think that VopS is just binding to the G protein. We think it has some catalytic activity, and can we use this assay? And so what we know as biochemists that enzymes work in a concentration and time-dependent manner. So can we use this assay to test whether VopS is catalytic? And so what we did is we took VopS and we added decreasing concentrations of VopS to this molecule rack, and we asked whether the key could fit in the lock. And what we observed was if we did the assay for like 15 minutes with a small amount of VopS, we decreased the amount of key that could fit in the lock, right? But this was only for 15 minutes, right? But if it's an enzyme and we do that same reaction and we do it for a longer period of time, then the enzyme should turn over the substrate, and eventually we should no longer see that rack can bind to pack, or we have a band on a gel. And so we did that assay, and that's exactly what happened. When we took that small amount of VopS and we incubated it with its substrate, over time, eventually we no longer could see the key fitting into the lock, which means that VopS was taking that as a substrate and turning it over, and it probably had a catalytic mechanism and supported our hypothesis. So what I've told you is in normal cells, rack normally can fit into pack, and it can make this actin cytoskeleton. But if VopS is around, this no longer can happen. And it is probably doing something catalytically to rack. And so what we decided to do was try to figure out what that was. And we did this in a similar way that I had told you before. We determined the molecular mass of this protein that had been incubated with VopS. And we got an amazing result. So what we found is that this rack molecule and the other G proteins were modified with AMP. And what's AMP? AMP comes from ATP. And ATP, of course, is a building block for nucleotides, and it's a reservoir for energy in our cells. And it's a precursor for that phosphate that was put on the signaling molecule that I talked about in the first story, right? It's the gamma phosphate of ATP that's added. But what we were observing is that the front half of ATP was being added to a molecule. And no, this was just unbelievable. Nobody had actually heard of this before. And so to put it in context molecularly, what we were observing is that normally what people had seen before with ATP is that the gamma phosphate could be used to modify a protein with phosphorylation. But what we had discovered was that this molecule that's been studied for decades has an entirely new function. And this new function is that AMP, the front part of ATP, can be added to a protein, modify the protein, and change the protein's activity. And this was just an amazing result. We were so excited about this. And we discovered first that this AMP could be added to a threonine. Then we showed it could be added to a serine. And then other people started studying other Fic domains and another type of enzyme that I'll tell you about in a minute. And this AMP could be added to tyrosines. And that's the same repertoire of amino acids that the phosphate is added to. And so this was an extremely exciting result because it was a new switch that no one had ever heard about before. But what I have to tell you is that 40 years ago, there was an enzymologist at NIH, Earl Statman, and he was studying a single bacterial protein that was involved in nitrogen sensing. And as far as he knew, it was the only example that he had ever observed of an enzyme that had been regulated by AMP modification on a tyrosine residue. So once before in science, this had been seen, but no one ever thought it was a general mechanism that could be used for signaling and modulating proteins. And so based on those studies that he had seen, he thought it was only isolated to this one bacterial enzyme. But what we did, we in a sense rediscovered what he had discovered and found a whole new set of enzymes that could carry out this type of modification, and that it's probably used in man, flies, and worms, because remember I told you there's Fic domains in those. And then we found a whole nother set of enzymes that look like the one that Earl Statman had been working on that can carry out this modification. So there's essentially a family of enzymes that can carry out this new type of modification, and now we're trying to study it in our own, in eukaryotic cells and to see what this mechanism is working on. So taking it, so this is the whole idea of why we do this. This is a model of discovery. These bacteria basically target the Achilles heel of a signaling pathway, and they use very unique mechanisms to do this, and they've been evolving for billions of years to try to manipulate the signaling systems in our body. And if we can figure out what they figured out a long time ago, we're going to find out a whole bunch of new things about our own body, and there's tons of these effectors more to study. So what I told you about today is that we discovered a new mechanism that involves the acetylation of serine and threonine residues and basically manipulates signaling pathways in cells, and that we found another new post-translational modification, what we call AMPylation, where AMP is added to proteins and it modifies the activity of these proteins. And I want to thank a lot of people. We have a lot of collaborators at UT Southwestern because actually with the pathogens, we never know what kind of pathway we're going to end up moving into, and we have a lot of experts there. So we have a lot of really nice collaborators. And I was hired there as a W.W. Caruth biomedical scholar. I've received support from the Beckman Foundation and an enormous amount of support from another Texas foundation, the Welch Foundation, which I'm very appreciative of, and the Burroughs Welcome Fund. And then we also receive help from NIH, and I'm hoping that that NIH support can be maintained in future decades to come. So thank you very much. Oh, you can't see it, forget it. That's great. It's so dark. Yes, sir?
Yeah, I have a quick, which one? Quick question. I can certainly understand the first mechanism, turning off inflammatory responses, being useful for the bacteria. But what about the second one?
Oh, I'm sorry. I probably didn't explain that. Yeah. So basically when the AMP is put on, the G protein, the key can no longer fit in the lock.
No, I meant the cells. The cells are now dying. I can understand why you turn off the inflammatory component in the macrophages and they're no longer
you mean
affected.
Globally?
From a standpoint of pathogenesis, that's very useful to the bacteria. But they must have evolved this other mechanism for some reason, and I didn't quite pick up on that. So it's a good question. So when you look at the model of the way the bacteria work, there's a number of effectors that are injected in. And so what I described to you is one mechanism out of probably a dozen for each. And all of these work together to basically induce a cell death. So on its own, it can induce cell death, but it actually works with a repertoire of other effectors to cause the cell to die. And actually, this mechanism, the second one that you were talking about, is actually really cool. Because what we think happens, so what we think happens is that Vibrio uses this to gain nutrients from the host. It kills the cell in a very particular way where it basically causes the cell to first eat itself, and then the cell rounds up, and then the cell bursts, and we think it releases nutrients.